So basically, we go through this, we refine the order as much as possible based on the diagnostics, we recompute the map with the chosen max offset, which was, and we end up with a map that we then want to validate against other maps of some sort. In this case, we validate it against the true map that we used for simulation, but in other cases it might be against some other published map, some consensus map, physical map if you have that. And for that, we use a function called mapComp. So it basically just compares two input maps by reducing them to the common markers. So it's matching up markers which have the same names. You also have to have the same chromosome names, so you just have to be aware of that. And you can apply it to either the maps themselves or NP-cross objects. What it does then is it picks out the markers on each map. It says, okay, do any of them have duplicate markers? Because that does happen. How many markers are there in common? In this case, because we're comparing the true map with the new map we've generated, you have the same number of markers, they're all in common, so that's fine. And it tells you how many markers ended up on different chromosomes in the two maps. So here I've already reset everything so that you end up with, I've renamed everything so the chromosomes in the original map are the same as the ones in the new map. And then it calculates, to start with, the correlations between the chromosomes. So you can see here they're all quite high, but two of them are negative, indicating that the chromosomes are flipped. And then it produces essentially that same plot I showed you from ggplot, although this one actually does it in lattice because I wrote it a while ago. And you can see here it's pretty much all along the diagonal. The lengths of the chromosomes are very similar. So you can see here this goes up to 100 on the left, and it goes up to 100 here roughly. But often you'll see quite different map lengths between whatever your validation map was and whatever you've generated. Okay, so that's all I'm going to say. That took a little bit longer than I was expecting. There are some exercises there. So there's two data sets. There's one which just has a map, that a data set that I've generated, and it was just to give you an idea of what the data set with the maps looks like, to try estimating recombination fractions and doing some of these plots, and seeing if you can guess how I generated the data as well. So the map's there, and if you can figure out what the commands were that I used to simulate the data, you can regenerate it yourself. You should be getting very similar things. The other one is a data set where I've messed up the map, essentially. I've moved things around, and it's up to you to try and correct it. Okay. So if anyone has questions about things that I've talked about, I'm happy to answer them. Otherwise, if you just want to play with the data sets for a bit, go ahead, or take a break, go to the bathroom, whatever. Yep. Yep. So if we go up here, well, I mean, there's one line of code. I mean, I don't know what you mean by simple trick. One way to do it, if you have the interactive tool installed, is to go in and just reverse it. That's very easy to do. The other one to do is you actually, you just subset the markers, essentially. So this object called sub, if we do sub is subset of itself, and then we say markers equals the number of markers, which is the number of columns in the object, the number of columns in the finals component, and we flip it around. That'll reverse the order. It won't re-estimate the map for you, but you can actually reset the map. You can basically subtract everything from the maximum, and that'll produce the flip of the positions as well. So in this case, I'm not sure it has a map. Yeah, so let me just do it for a different one. Okay, so for this one, if we wanted to flip chromosome 1, for example, I'm going to make this bigger. So chromosome 1, this is the original map, so we know exactly what order, but if we wanted to flip it around, then we can set the map to to basically, what we're doing is we're subtracting everything from the maximum value and then just ordering them again, and that will flip everything around. That's what that command does. There's not a function to do it. I guess that's the answer, but it's a relatively simple command, and so that now is taking the one at the end of the chromosome and setting it to 0 and so on, and then you just reorder the object essentially based on that order. As long as you do have to subtract the values from the maximum, otherwise what it'll do, the position of the starting marker would be 100 instead of 0.
I know I already used one time the question with the criterion under empty order does not produce this.
Oh, really? So it's not a function, it's an argument. If you do,
So that's in the error message, but yeah, I mean, we pulled up the help file and tried to exist criterion, it's not exist. Are you glad you have all these new people to troubleshoot?
Could you do it without?
Well, we tried that before.
Oh, you tried that.
See, that was the first one, which is how you had it written. And it keeps giving me this error. So then we went and looked at the help and thought, well, maybe we need to put the other stuff in there.
Yeah, no, you shouldn't need that. So it's definitely, it's making me wonder if there's a different version installed on
it seems like
your
it's a
computer
different
than
version.
mine, because obviously that worked on mine.
But the help file says criterion.
Yeah, no, it should definitely be in there. I'm guessing there's something in the code that's going wrong. Let me try one thing. This is the problem when you don't have a PC to run things on, I guess.
Oh, that's weird. If I delete criterion, it still says cannot find function criterion.
Yeah, if you put in a function, it'll just pull up all the code. It's getting down to the point where it starts to order things. Oh, I wonder if... So,
Misspelled somewhere.
no, I wonder if you actually have almost a newer version than mine. I'm wondering if, so Rowan, the student who works with me, I wonder if he's made a change recently, and you actually have a newer version than I do, and that's broken something, because that, I mean, that's a core, I'd be surprised if he changed it, but.
If missing criterion, and so I just submit.
Yeah, so it gets past that because it prints out ordering chromosome 1. So further down, you'll see where it kind of says, you know, yeah, cat paste down there, ordering chromosome. So it's past that when it fails. It's probably when it's feeding into one of the C chunks.
The C chunk?
Yeah, so this function NP order chunk, I can't see where that is. It might be further down. Yeah, that one, NP order chunk. that actually calls some C code. Just here. And I'm wondering if it's when it's getting fed in there that something's going wrong.
Yeah, I don't know. Let's see.
Yeah, I'll have to take a look at it. It looks like there's a bug. Yeah, no, it's just odd because I looked at the version that was installed on one of these and it matches what's mine. It's just a question of whether he's actually documented a change he made. So,
Okay.
Sorry about that.
No.
Okay, I'm going to talk for about 15 minutes on QTL mapping now. As it turns out, I didn't realize we're having a break at 10, so I might as well just get started at least. So basically, I just want to go through briefly the models that I talked about in my talk on Tuesday. The full model is the main one we use, which is based off of estimating different effects for each of the founders on your trait. Association mapping, which is just based on estimating the effect of your observed alleles, and then the meta-alleles, which is using something in between those two. I've put mixed models in here because you can use MP-map to do that, but you have to have ASVML installed to fit the mixed models, and it's not something I'm going to go into in a lot of detail because it could take a long time to talk about mixed models themselves. So the starting point is for the QTL mapping is always going to be the genetic map, basically. So in this case, this is the genetic map produced from the previous stage. You can see that some of the chromosomes are shorter than they really should be. One is a bit longer, but roughly they're about right. And the goal is to associate some sort of phenotype with your genotype. So this is just a plot, much like the ones Carl showed you, of a QTL profile. So you have, in this case, rather than plotting the LOD score, plotting the negative log base 10 of the p-value. And we're just trying to pick up QTL on various chromosomes. Before anything else, the first question to ask yourself is, what does your trait look like? Is it normally distributed? Do you have transgressive segregation? Do you have more than one trait? Do you have more than one field you're looking at? Are you looking at it over more than 1 year? All of this is going to play into how you analyze the data. And so, for example, this is one of our traits down here where the colored lines are indicating the mean values of our founders. So this is from the 4-parent population. And the histogram is showing the values in the progeny. And you can see that the 4 founders are very far to the left, and quite over half the data basically is above those 4 founders. So we have quite a bit of transgressive segregation. And that's true for a large number of the traits we see. It is fairly normally distributed though, and so the approaches which are built into MP-map will work relatively well. If you have a binary trait, they won't work that well. So beyond what I'm going to talk about, and really beyond what's in the base MP-map, are a number of things. One is epistasis. If you want to look for biallelic gene-gene interactions, I recommend looking at these other 2 packages, EpiGPU or GLIDE. They were built for human association mapping studies, but they will work for many other studies. And I've put references at the end of these slides. If you want to look at the full model or anything else, then there's a collaborator at CSIRO, Josh Bowden, who's working on an R package which can be used generally for multi-parental populations. In terms of binary traits, while they're not too difficult to modify for linear models, they're not built into MP-map at the moment. And they are more complex for mixed models. So I have some collaborators at CSIRO who have fit mixed models with binary traits, and they published that earlier this year, but it's not a simple process. And then for multiple traits, in theory, you can fit it with MP-map using the mixed model capability, but you may want to consider other approaches, and that's another, MPW game there is another approach which was published by some collaborators at CSIRO again, where they were looking at a mixed model approach for multiple traits in multi-parental populations. And the upshot of it, I guess, is that it works pretty well, but it takes a very long time to fit, and you have to be very careful about the types of models you fit. So it's very computationally intensive, and it's quite complicated. The other thing you can do is try and combine univariate analyses, which may provide you just as much power, although it may not provide you quite as much information as you want. So what is in MPMAP are the models that I talked about. So the full model, or linkage, was originally proposed by Richard Mott. And the idea is to basically specify that your trait Y is affected by each of the founders with any possible combination of traits. So here, your beta Fs are different effects of each of your founders on your trait. So your first founder might have a positive effect on yield, another founder might have no effect, another founder might have a negative effect. And so you're allowing those values to change between all your founders, you're giving it full flexibility and estimating all the founder effects. The possibility is that there's too many there, really, because it may be that 3 of the founders have no effect. In order to fit this model, what we use are the identical by descent haplotypes. So we're looking at regions of the genome which were inherited as a chunk, and the full model is trying to capture the effects of inheriting each chunk from different parents. We estimate which alleles are inherited from the different founders using a hidden Markov model, which depends on the distance between markers, the genotypes of the founders, the genotypes of the finals, and the population design. So basically all of the information that we're putting into our MP cross objects is used to calculate these genotype probabilities. And if your pedigree changes, that will also change these probabilities. So I showed this diagram on Tuesday, so I'm not going to go into it, but essentially what we're trying to do is pull apart these chunks, and we're trying to do it with as much confidence as possible. The function we use to do that is called MP-prob, and basically you feed in your MP cross object. You choose a program, so you can either compute it with RQTL, so using exactly the same machinery as the calc genoprob function in RQTL, or you can estimate it using HAPI, which doesn't use any of the pedigree information, but also produces probabilities, and then you choose a step size. So in this case, it's choosing markers every 2 centimorgans. One of the things Carl mentioned yesterday was a function called reduce to grid, which after you've estimated the probabilities, reduces down to just that grid that you've specified rather than including all the markers. You can do the same thing in this function by setting an argument called MRK-POS. And that's just saying, so that's meant to stand for marker positions. If you set it to true, then it includes all of the marker positions. If you set it to false, it takes them out. So this setting is equivalent to reduce to grid in RQTL. And what that means is that even if you use this setting, even if you have 1,000 markers on a chromosome, most likely the limit of the number of positions you're going to look at for your QTL mapping is maybe every 1 centimorgan. So then you're dealing with, say, 200 or 300 different positions rather than all 1,000 markers. If we look at some of the probabilities that we get out of this, basically everything which is output from MP-prob is stored as a list called prob in your object. So within each list, you have one component for each chromosome. That's what this is for. And then each of those is a matrix where the rows are the number of lines and the columns are the number of markers multiplied by the number of founders. So in this case, we have 4 founders, and I'm just printing out the first 2 markers and the probability values for the first 3 lines. So as you go across, you can see you have a group of 4 founders. For the first line, at the first marker, you have a very high probability of inheriting the allele from founder 2. For the second and third lines, you have a very high probability of inheriting the allele from founder 1. Similarly, at marker 2, you have basically the same pattern. And so there's another component which is generated from this called ESTFND, the estimated founders. And what that does is it basically goes across and says, which of the founders are you most likely to inherit an allele from? So it's trying to actually compress that probability information down to the most likely founder. And this information is useful in terms of looking for crossover events, because if you're going from one founder to another, obviously, it's likely that a combination occurred there. So if you're going across and you see 2, 2, 2, 2, 2, 3, 3, 3, then some sort of crossover occurred at that point. As far as the biallelic model goes, that's much simpler. We're just estimating one effect, essentially, for each marker. The drawback, of course, is that if you have multiple founder effects, you're unlikely to be able to pick them up. But it's much more computationally simple, and it's much faster, and there are a lot of different software packages you can use, actually. The grouped model, then, is trying to do something in between. So I showed you some citations for ClusTableau previously. I'm presenting 2 alternate approaches next week, and we've done some simulations testing out them and basically trying to determine the best way to construct groups. You can do it any way you like, really, but ideally you have some consistency with the observed alleles and you have something which is biologically meaningful. So all of these are implemented in MPMAP, and so I'll show you what they look like in terms of the Arabidopsis magic data. So one of the data sets which is on GitHub is this Arabidopsis example. It's the data that's available on Richard Mott's website, which was from the original paper by Cover et al. In 2009. And so that was from the Arabidopsis magic population, 19 founders, 1,254 SNPs. And I've already gone ahead and grouped the alleles according to one of the schemes I presented on Tuesday. So this vector of the number of alleles is the number of alleles at each marker for this grouped model. The probabilities for this data set have already been happy. So this is an MP-prob object, it should be. We can look at the distribution of the days to bolting, which is essentially flowering time, and get an idea of what we're dealing with. You can see it's skewed. It's not quite normally distributed, but it's a quantitative trait. It will probably work reasonably well. And then in order to actually do QTL mapping, the main command is called MPIM. We use this whether you're fitting any of those models, whether you're doing mixed model interval mapping, whether you're doing composite interval mapping. It's just a matter of changing the arguments there. So the main argument is you have to put in an object. It can be an MP cross object or can be one where you've already computed the probabilities. You put in the number of covariates you want to include, so that determines whether you're doing interval mapping or composite interval mapping. And you also need a response name in case you have multiple phenotypes. So the full model, you just put that information in, essentially. For the biallelic and the grouped model, you need another argument which is called founder groups. And so in the case of the biallelic model, your founder groups are your observed genotypes. So you just put in the genotypes of the founders. For your grouped model, you put in whatever those groups are. And then, if you produce the results, these objects are now called MPQTL objects. So we gave them a different class because then we can do different summaries and produce different plots by default. And basically it summarizes the QTL that you find afterwards. When you plot it, you plot the QTL profile, and you can plot the support interval as well. So if we look at the summary of the MPQTL object for the full model, this finds something with 43 QTL. That seems like a lot. The reason for that is that by default, when it finds the peaks of the QTL profile, it's looking on a very fine scale. Now, we know that if we have QTL which are detected, if we have peaks in our plot which are right next to each other, it's unlikely that they're separate QTL. And so what I've done here is refine it by basically making a more smoothed QTL profile. So this D window argument will smooth the profile slightly so you're not picking up every little blip in the graph, basically. And so find QTL is the function which we use to basically tweak how we're defining the QTL peaks. I've also changed the threshold here. So that's a negative log base 10 p-value of 6, which means that p-values have to be below 10 to the minus 6 in order to pick it up. So that's quite a conservative threshold, but that was just to narrow things down so it's easier to look at the results. And you can do that for all of the things. Typically, the D window, I'd actually recommend something a bit higher. So 5 corresponds to essentially smoothing over a window of 0.5 centimorgans. So 50 would be about 5 centimorgans. And you're unlikely to be picking up QTL which are within 5 centimorgans of each other anyway. So this is a plot of all 3 of the results there for one of the chromosomes. And as I mentioned earlier, these 2 peaks correspond to 2 genes known to affect flowering time in Arabidopsis, FRI and GA. And if you actually output the summary, which I will do if I can get this to load up. Okay. So I did not put the file results Arabidopsis on GitHub because it's a very large file. But if you run basically those mpim commands, that's what it should produce. They take a little while to run, but you could set it running. We're about to have a break, so you could set it running now, and then you should be able to look at the results by the time that we come back. So if we look at, for example, the summary here, it prints out a table where for each QTL, it gives you information on the chromosome, the position on that chromosome where you're detecting it, the flanking markers, the Wald score, the test statistic there, the degrees of freedom of the test. In this case, it's 18 because we're testing all possible combinations, and the p-value. This is the output where I haven't restricted the p-value, obviously, because you're getting things which are greater than 10 to the minus 6. But you can see in this case, because I haven't smoothed the profile, you're getting a huge number of QTL, most of which are just due to little ups and downs in the QTL profile. If we plot it, then again, these green bands indicate the support intervals for different QTL. Because there's way too many QTL there, and I'm just going to do this. To make it easier to see. Yeah, so if we basically, you see all of these little peaks here, all of these little ups and downs. That's what's being picked up if you don't set the D window to a higher value. Whereas if you smooth it, you only pick up this one peak on chromosome 1, this very large one on chromosome 4, and some large ones on chromosome 5 as well. But basically, the default plot is just to show all of the chromosomes, show the QTL profile, and show the support intervals for the QTL which have been found. And if we summarize this one, as I said, there's only the 3 QTL now. It's picking up a SNP in the gene FRI on chromosome 4, and it's picking up SNPs on other chromosomes, which are highly significant in this case. Very large effects. For this Arabidopsis data, it's not outputting the founder effect estimates because it has 19 founders, so it's not set up to do that. For 4-parent and 8-parent causes, it would also output the estimates and their standard errors. And I think that's a good place to stop. We're going to have a break. If people are trying to run MPIM during the break, then let me know if you run into problems.